Well, we could we'll have to talk about probably things about metabolic reactions as they come up.
Okay.
All right, Beth.
Yep, let's do this.
Let's do it.
Wait, I have to get my okay. So a lot like Lindsay's, this presentation is like an introduction to like cell signaling and then specifically the role of RAS in signaling pathways. But like I was saying, like a lot like Lindsay's, it's more of like the biology side of biochemistry. So I have like a brief introduction in the beginning. But I put this here because I think it's cool. And this is a lot of all of the interconnected signaling pathways involved in apoptosis, which is programmed cell death, gene regulation, which is really like a blanket term for basically all of the central dogma of biology, so DNA to RNA to proteins, and cell proliferation. And I think it's just really interesting to see how interconnected all of these processes are. What I'm going to be focusing on specifically is this one right here. As you can see, there's RAS, and this is the pathway that a lot of what I've been doing research on focuses on, and it ties directly into gene regulation. But a bunch of other interesting things are like here's P53, which is involved, which is a tumor suppressor gene, and if it's turned off, like if it's non-functional, it can lead to cancers. And then CDK2 and CDK4 cyclin-dependent kinases are cell cycle regulators. So if they are mutated or messed up, they can also lead to cancer. So it's just interesting to see like how one little mutation in one part of these can lead to like such detrimental effects, which is what I'm going to be like specifically focusing on. So I have like a brief overview here of like what's in the presentation because it's kind of like a lot. So the first is like introduction to what cell signaling is and signal transduction pathways and the steps involved and like how chemical messages are sent. Then what is RAS and why is it like so notorious in the scientific community? The pathway RAS is involved in, also called the MAPK pathway, and then the role of RAS within that pathway as a signaling protein. And finally, the main reason why RAS is studied so much is because of its mutations, which eventually lead to cancer. So cell signaling, as the name suggests, is a form of cell-to-cell communication in which a sending cell can send out these ligands, which are just like chemical messages. Ligands?
Ligand is how biologists pronounce that word. So, I mean, I'm not going to like go to the mat over this, but like most chemists pronounce it as ligand and biologists pronounce it as ligand. So I would prefer that you pronounce it as ligand, if that's okay.
Okay, yeah, well, that's how Janet
this
said
is really nice.
All my bio professors call it ligand, so I didn't know.
I know. Yeah. It's a struggle. We're all fighting our own fight for our own team. You know what I mean?
Okay, so it's a ligand. Yeah. But they are chemical messages that can be sent and received by a target cell. So as you can see, this target cell has a receptor right here that is specific for this ligand, and it can bind and ultimately transmit a response. But if you see this cell over here has no membrane-bound receptor, so it can't receive that signal. So it is selective. And there are four main types of cell signaling: paracrine, autocrine, endocrine, and synaptic. Paracrine signaling is like what's indicated right here. It's short distances, so cells within the same area can kind of speak to each other. Autocrine is just like the prefix suggests. It's this sending cell and the target cell are the same cell. So it can send out its own ligands, target itself, and elicit a response within itself. Endocrine signaling is more long distance signaling, like the endocrine system. So if one part of your body wanted to communicate with another part of your body, that would be endocrine signaling, and it normally travels through the bloodstream. And then synaptic signaling is like in your neurons, like in your brain. They're like synapses, that's where it kind of comes from, so neurotransmitters. And then what I'm going to be going into next is the three main steps of how this process works. So the first step is really simple. Ligand binds to receptor, and there are multiple types of these receptors. So intracellular are ones inside the cell, and the ligands that bind to these are small nonpolar molecules that are able to get through this lipid membrane. So like estradiol and testosterone are ones that can like actually get through the membrane and bind to a receptor inside the cell. The most common are cell surface receptors, which is just like this one illustrated here. So the ligand bonds to the portion outside of the cell and elicits a response inside the cell, so it doesn't actually have to go through the membrane. Then there are ligand-gated ion channels. These are mostly found like in your neurons. So a neurotransmitter can bind to these ion channels and allow a potential, like an ion potential, to elicit like responses like that, like how your neurons fire. And there are G protein coupled receptors and RTKs, and I put a giant exclamation point by these because this is what's important in RAS. So RTKs are receptor tyrosine kinases. So I have a little bit of a comic here as to like how these work. So step one, signal molecule binds to binding site, and you can see that these are like the monomers, and they're normally like in pairs of two, but they're separated because they're not activated quite yet. So I have an illustration like the structure of tyrosine. That's what it looks like, and there's, right, apparently there's 6 right here. So when 2 of these signal molecules are able to bind to each receptor, they dimerize, they come and stick together, and that activates them. And then after these are activated and the ligand is bound, ATP phosphorylates these tyrosines. So ATP, like Lindsay mentioned, is essentially energy. It's adenosine triphosphate, looks like this. And when it gives up a phosphate, it becomes ADP, adenosine diphosphate. So like just cut one of these off. And that's what a phosphorylated tyrosine looks like. Oh, and just as like a side note, kinases slap phosphates onto things. That's what their job is.
Correct. That's right. That is correct.
So it is able to phosphorylate itself, and it's become like fully activated. And then these inactive proteins, which want these phosphates to activate themselves in the rest of the signaling pathway, are able to take them and elicit response down a relay chain of transduction. So this is essentially like the first step of the RAS-RAF pathway. So step 2 in this process is transduction. So it causes so the ligand binding to the receptor causes a conformational change in that protein, which turns it on. It tells the rest of the cell that like there's a signal, and then a cascade of proteins can transduce that signal and amplify it until that response is reached. So another little comment here, the pathway is off because no ligand is bound. Once it is bound, it's able to start this cascade of turning every protein in the pathway on. So it's like a game of telephone. So they pass the same message down through each other, except in this game of telephone, the message doesn't get mixed up. So it just stays the same. And it's able to elicit the target response in the end.
Did you make these?
No, I stole them from Khan Academy.
Oh, okay. Yeah, these are very nice.
Yeah, I'm not that talented. So there are 2 types of general responses that can be garnered by signal transduction pathways. The first one is gene expression, which, like I said earlier, is like a blanket term, DNA to RNA to protein. So you can turn on the transcription of certain genes, which can then be translated to make those proteins that do that certain function that might be needed in the cell. And this could be problematic if they're turned on all the time, like in cancer. And another one is cellular metabolism. So this takes place in the liver, and a molecule of epinephrine binds to its epinephrine receptor, and it turns on adenylyl cyclase, which is really interesting. It takes adenosine triphosphate and turns it into CAMP, which is cyclic adenosine monophosphate. So it has like one phosphate group, but it's like cyclized when it's bound to that adenosine group. And CAMP is a secondary messenger, so it's able to do 2 functions. It can first turn off this glycogen synthase, so no new glycogen is made, and activate glycogen phosphorylase, which breaks glycogen down into glucose, which pulls up glucose in your liver and is what elicits that fight or flight response. So it can act in this metabolic way as well. So those are just 2 examples of like the type of responses you can get through these pathways. Next. So now we can actually talk about what RAS is. So RAS is a superfamily of a bunch of related proteins, all belonging to the small GTPase class. And these small GTPases are just able to hydrolyze GTP, which is guanosine triphosphate. So similar to adenosine triphosphate, just different nitrogenous base, into GDP. So it's able to take that phosphate group off. And they are obviously involved in signal transduction, and they communicate binary messages. So they're either on or they're off, and they can pass that downstream to other proteins. And then what's interesting about them is that HRAS, NRAS, and KRAS are all oncogenes, which you can think of like the gas in a car. So if they're like heightened up, it's like you're accelerating and you're like constantly causing cell division, which builds up into cancer and tumors. And then over here, I have the structure of activated HRAS. So activated means it's bound to GDP. So here's that guanosine group right there. And then there's the 3 phosphates in orange. And then this green dot right there, that's a magnesium 2+ ion that's like also bound in there like we learned in quant. So that's really cool. And then these purple arrows are the beta pleated sheets of the protein structure. And then the teal spirals are alpha helices. And then these gray like strings are called loops. And that's what connects them. So I did try making this. So this is the RAS, RAF, MEK, ERK pathway, also called the MAPK pathway. And this is where RAS is. I indicated that by calling it the problem child because it is. And so these are the receptors, TGF alpha, which is transcription growth factor alpha, and then EGF, which is epidermal growth factor. And EGF binds naturally to EGFR, epidermal growth factor receptor. And it starts this whole pathway. So growth factor receptor bound protein 2 is bound to the growth factor receptor. So that's actually pretty self-explanatory. Son of sevenless or SOS. There's an interesting story on Wikipedia as to why it's called this, but I'm not going to get into that. It's a GEF, a guanine nucleotide exchange factor, which I'll talk about in the next slide. But essentially what it's able to do is tell RAS, hey, it's time to give up your GDP, take your GTP, and like turn the pathway on so it's activated. RAS activates BRAF and RAF1, which are also kind of problematic because they can be mutated as well, which activates MEK1 and 2, which MEK stands for MAPK/ERK kinase. And what's interesting that I learned is MAPK and ERK are used interchangeably. MAPK is mitogen activated protein kinase. Again, kinase just means slapping phosphates onto things. And ERK is the extracellular receptor kinase. So they're used interchangeably. So sometimes you'll see ERK in pathways also called MAPK. And so MEK activates ERK, and ERK translocates into the nucleus and does all this cool stuff. But what's most important is these transcription factors, specifically FOS and JUN, which are able to activate transcription for genes involved in cellular proliferation and growth. And then what's also interesting is here's translation, so RNA to proteins, and acetylation, which is what opens up DNA that's coiled around histones, so it can be transcribed and can be duplicated in the synthesis phase of the cell cycle. So that's essentially how the pathway works, and this is how RAS works. So here's GEF, so that's your SOS protein. So it is turned on, and RAS is inactive right now. It gives up its GDP, switches it out for GTP, and it's able to pass a signal downstream to RAF. So now it's activated. And then a GAP, which is a GTPase activating protein, tells RAS to do its job, get rid of a phosphate, hydrolyze it back down to GDP, and turn itself off. So it's a good little kind of feedback loop here of RAS turning on, being turned to told off. So as you can see, if you mess this up, RAS can stay always on. And that's what happens. So mutated RAS is unable to hydrolyze GTP, its on form, back to GDP, its off form. So it's always on and always translating that signal downstream, telling them like transcribe more genes, make the cell bigger, make it divide more. And most mutations, which is really interesting, occurs at position 12, 13, or 61 in either of these 3 isoforms. So like those are like the problematic positions for where the amino acids are located. And so I found this really cool diagram here of a KRAS codon in chromosome 12. So normally the DNA code is GGC, which codes for glycine, and that allows RAS to do like normal function. So it can go from its inactive phase to its active phase back to inactive. So everything grows and divides as normal. But if it's mutated, and this is called a point mutation, if one of these little codons is switched to a T instead of a G, you get valine or valine, I don't know how to say it, and your inactive form is not reached. So RAS always stays activated. It essentially makes GTP like kind of lock in there and not able to leave. So it's always on, and it causes abnormal growth, proliferation, and differentiation, otherwise known as cancer. So this is why RAF is so notorious and why people want to study it and target it so much, is that more than 30% of human cancers are caused by mutated RAS genes. And if you think about that, it's kind of crazy because more than 30% of cancer is caused by one gene at these 3 positions, basically. So it's so minute, but it can cause so much damage. And of these 30% of cancers, 95% of all pancreatic cancers are caused by RAS mutations, 45% of all colorectal, 35% of all lung, and a lot more. The list went on. So here's like a diagram from the American Association for Cancer Research, and it shows these three positions, 12, 13, and 61, in HRAS, KRAS, and NRAS, and how common they are in determining like what cancers are caused by them. So if you look at KRAS and pancreatic cancer, almost 100% of the time it's caused by a mutation in position 12 in KRAS. So, and then you can look over here for colorectal. Position 12 in HRAS, 100% of the time would cause colorectal. So it's really interesting to see the differentiation between 3 positions and 3 different isoforms of RAS. And lastly, I just wanted to go over that this pathway in general is kind of problematic for mutations. So you could have a mutation in EGFR or overexpression, RAS mutations like we went over, and BRAF mutations, which is another interesting point of discovery for targets. But what I wanted to focus on, like real quickly, I put a little star here. Lindsay would know this from our talk with Maki. Now that I understand a little bit more about this pathway, she had non-small cell lung cancer, which was caused by an EGFR mutation. So this is eventually what caused her cancer. And then she had a further mutation when she started taking Tarceva, which resulted in a point mutation within EGFR, allowed the original drug not to be able to bind to that pocket anymore, causing her to switch to Tagrisso, which is specifically designed for that one point mutation in that one receptor in order to allow it to bind. So that's like precision medicine. That's an example of like how that works. So I just think it's interesting that this entire pathway can be changed by like one point mutation in one aspect of it. So that's just like an overview of what I'll actually be getting into. Hopefully I can talk more about how they're actually targeting RAS and like the chemistry behind that, but this is just why RAS is so important in cellular function. So questions?
Questions. Thank you, Beth. Yeah, it's interesting that you brought up Maki's cancer, right? She's really open about sharing like the details of what the biochemical cause is. And I think you're right to point out that like almost every member in this pathway, with maybe the exception of GRAB2 and SOS, there are mutations in them that can drive the formation or the progression of tumors, including EGFR itself. So all of these are anti-cancer drug targets. Like you said, EGFR, there's Tarceva, which was the sort of the first line drug that inhibits EGFR. She's eventually going to talk about RAS drugs, which are brand new, which is like probably the most exciting thing to happen in cancer biology for decades, and so on. So it's good. It's also nice, I think, that both of you, Lindsay and Beth, have both talked about kinases. And kinase is a old school name for enzymes that are phosphotransferases. So they'll take a phosphate group from one molecule, typically ATP, and transfer that phosphate to another molecule. So in Lindsay's case, the recipient of the phosphate is the metabolite, right? So shikimate kinase phosphorylates shikimate with ATP. In your case, Beth, all of your kinases are called protein kinases, where the molecule that gets phosphorylated is a protein, and then in the signaling context, that turns that protein on. So like RAF and MEK and ERK are all protein kinases that are activated by phosphorylation and then in turn phosphorylate other things, right? So that's good. Yeah. So do you all have any questions for Beth? It's just silence. people like not eat breakfast, this hour change is just